dear students. I am Pratima Sharma, your bio teacher from St. Jude's Academy, Sonipat. Welcome to Digital Bio Learning. In the first session of your first chapter, the fundamental units of life, we have already covered some topics like what are living organisms made up of? How was the cell discovered? Who discovered the cell? Why is the cell called the structural and functional unit of life? In the structural organization of a cell, we discussed about some of the cell organelles like the plasma membrane, its characteristic, the selective permeability. Now, what all things it lets inside the cell, what all things it lets outside the cell.
We have studied about the difference between the hypotonic, hypertonic and isotonic solutions and their effects on the cell membrane. The next cell feature which we'll be studying would be the nucleus. The nucleus has a double-layered covering called the nuclear membrane. The nuclear membrane has pores which allow the transfer of materials from inside the nucleus to its outer side, that is to the cytoplasm. The nucleus contains chromosomes which are visible as rod-shaped structures when the cell is about to divide. And when the cell is not dividing, at that time it is present in the form of chromatin material, which is a kind of entangled mass of thread like structures, right? Whenever the cell is about to divide, the chromatin materials get organized into chromosomes. Okay, now chromosomes contain information for the inheritance of features from parents to the next generation, that is to the offsprings, in the form of DNA, that is deoxyribonucleic acid molecules. Fine. The chromosomes are composed of DNAs as well as the proteins. Now, these DNA molecules contain the information which is necessary for constructing and organizing cells. Now, functional segments of DNA are called genes. I repeat, the functional segments of DNA are called genes. Now, in a cell which is not dividing, we already know that DNA is present in the form of chromatin materials. The nucleus plays a very central role in cellular reproduction, the process by which a single cell divides into two. It also plays a very crucial part along with the environment in determining the way the cell will develop and what forms it will exhibit at maturity by directing the chemical activities of the cell. Now, on the basis of the presence of the well-defined nuclear region and membrane-bound cell organelles, the cells are divided into two, that is the prokaryotes and the eukaryotes. The organisms whose cell lack a nuclear membrane are called prokaryotes. Now, pro means primitive or primary and karyote means carry-on, that is the nucleus. Okay, the organisms with cells having a nuclear membrane, a well-defined nuclear membrane, are called eukaryotes. In the prokaryote cells, they lack the other cytoplasmic organelles which are present in the eukaryotic cells. Many of the functions of such organelles are also performed by poorly organized parts of the cytoplasm, like for example, the chlorophyll in photosynthetic prokaryotic bacteria, right? Which has membranous vesicles but are not plastids, okay, like how we have plastids in the eukaryotic cells. Now, as we know, there are three features which are present in every cell. The first one is the plasma membrane, the second one is nucleus, and the third one is the cytoplasm. Okay, now what is cytoplasm? The cytoplasm is the fluid content which is inside the plasma membrane. Okay, now fluid content means it is the protoplasm plus the cell organelles. Okay, the cell organelles like Golgi apparatus, lysosomes, everything in the protoplasm both together are termed as cytoplasm. Now we shall discuss about some of the cell organelles, the main cell organelles, right? The first one would be the endoplasmic reticulum. The endoplasmic reticulum is a large network of membrane-bound tubes and sheets. It looks like long tubules or round or oblong bags like vesicles. The endoplasmic reticulum membrane is similar in structure to what we have it in the plasma membrane. Now, endoplasmic reticulum is also of two types, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough looks rough under the microscope because it has particles which are called ribosomes which are attached to its surface, because of which it seems rough. The ribosomes which are present in all active cells are the sites of, now what is the function of these ribosomes? Right, these are the sites of protein manufacture. The manufacture protein, the protein which is manufactured here are then sent to various places in the cell depending on the need. Using the endoplasmic reticulum, the smooth endoplasmic reticulum helps in the manufacture of fat molecules or the lipids, which are important for the cell functioning. Some of these proteins and lipids help in building the cell membrane also. Now, this process of building the cell membrane is known as membrane biogenesis. Some other proteins and lipids function as enzymes and hormones. Although the endoplasmic reticulum itself always forms a network system. Thus, the main function of the endoplasmic reticulum is to serve as channels for the transport of materials, that is, specially proteins, between the various regions of the cytoplasm or between the cytoplasm and the nucleus. The endoplasmic reticulum also functions as the cytoplasmic framework, providing a surface for the biochemical activities of the cell. In the liver cells of the groups of animals called the vertebrates, smooth endoplasmic reticulum plays a crucial role in detoxifying many poisons and drugs. The second cell organelle we'll be discussing is the Golgi apparatus. Golgi apparatus consists of a system of membrane-bound vesicles which are arranged parallel to each other in stacks, which are called cisterns. That is, the stacks in which parallel to each other they are arranged, right? These stacks are termed as cisterns. These membranes often have connections with the membranes of the endoplasmic reticulum and therefore constitute another portion of the complex cellular membrane system. The materials synthesized near the endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through this Golgi apparatus. Thus, the function of Golgi apparatus includes storage, modification and packaging of the products in vesicles. In some cases, complex sugars may be made from simple sugars in Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. Now, what are lysosomes? These are the third type of or the third kind of cell organelles. Lysosomes are a kind of waste disposal system of the cell. Lysosomes help to keep the cell clean by digesting any foreign material as well as the worn out cell organelles. Foreign materials entering the cell such as bacteria or food as well as old organelles which end up in the lysosomes, that is, which break them into smaller pieces. Actually, lysosomes are able to do this because they contain powerful digestive enzymes which are capable of breaking down all organic materials into simpler forms. Like during the disturbance in a cellular metabolism, for example, when the cell gets damaged, lysosome may burst and the enzymes which are present in the lysosome digest their own cell. Therefore, lysosomes are also called the suicidal bags of a cell. Structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. And these enzymes are made by the rough endoplasmic reticulums. The fourth cell organ we'll be discussing is the mitochondria. These are known as the powerhouses of the cell. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP. Now, ATP is adenosine triphosphate molecules. ATP is also known as the energy currency of the cell. The body uses energy stored in ATP for various metabolic activities. Mitochondria has two membrane coverings inside of just one. The outer membrane is very porous, while the inner membrane has deep folds in it. These folds actually gives a larger surface area for ATP generating chemical reactions. Mitochondria are strange organelles in the sense that they have their own DNA and their own ribosomes. Therefore, mitochondria are able to make their own specific proteins. Now, the fifth cell organelle we'll be discussing are the plastids. Plastids are present only in plant cell. There are two types of plastids, chromoplasts and leucoplast. Chromo, colored plastids. Leuco, white or colorless plastids. Plastids containing the pigment chlorophyll are known as chloroplast. Chloroplasts are important for photosynthesis in plants, that we already know, right? Chloroplasts are also sometimes contain various yellow or orange pigments in addition to chlorophyll. Leucoplasts are primarily organelles in which materials such as starch, oil, protein, granules are all stored. Now, the internal organization of plastids consists of numerous membrane layers embedded in a material which is called the stroma. Plastids are similar to mitochondria in the external structures, and they are also similar in one more way. Like mitochondria, plastids also have their own DNA and ribosomes. That means both mitochondria and plastids, they both have their own DNA and ribosomes. That is, they can prepare their own kind of proteins. The sixth organelle which we will be discussing are the vacuoles. Vacuoles are the storage sacs for solids and liquids contents of the cell. Vacuoles are small sized in animal cells, while in the plant cells they are quite large. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. Many substances like amino acids, sugars and various organic acids and some proteins also in plant cells are stored in these vacuoles. So, now we can say that each cell acquires its structure and ability to function because of the organization of its membrane and organelles in specific ways. The cell thus has a basic structural organization which helps the cell to perform its functions. So, the cell is the fundamental structural unit of living organisms. That is all we have in the fundamental unit of life. Thank you. Stay blessed.